So we start off. So wait. Right. So if this is his axial membrane, that's the outside. That's the inside. Right. Outside has comparatively more sodium ions than inside, and inside has comparatively more potassium ions. Because we've got more positively charged sodium ions on the outside is more positively charged in comparison to the inside, just because we've got more positive charged ions on the outside than the inside. Remember that you're always talking about the movement and relative movement of positively charged ions, not negatively charged ions, because there's no negatives moving. Right? So, your axial membrane, remember, we've got voltage-gated potassium channels and voltage-gated sodium channels. Right, so, the first thing that we need to think about, right, so at resting potential, the concentration of sodium ions outside of the axial membrane is higher than inside. Right, because we've got more positives on the outside than the inside is positively charged in comparison to the inside. So if the outside's comparatively positive, then the inside must be comparatively negative. Right, next thing that happens, right, so stage 2, so the potassium ions inside the axon are a lot higher than outside, right, so we need to remember that. It's always worth remembering that whenever we're talking about different numbers of ions or different numbers of anything being higher or lower, at some point we're going to be talking about diffusion, and whenever we talk about diffusion, talk about things moving down concentration gradients. So, the overall charge of positive ions is greater on the outside of the axon than the inside, making the outside relatively positively charged, making the inside relatively negatively charged. Right, remember that that's what we talked about when we were going through the maintenance of a resting potential. Right, so when we're at resting potential, the outside is positively charged and the inside is relatively negatively charged. And we've got that resting potential of minus 70 millivolts. So, the first real thing that happens in this propagation of an action potential is that a stimulus causes voltage-gated sodium channels to open up. Right, so now these sodium ions can go and start to go and diffuse into the axon. Right, so this causes a reversal in the charge because we end up now with more positives on the inside in comparison to the positives on the outside. So now the inside becomes relatively positive, so therefore the outside must become relatively negative. Right? So it's reversed, so it's flipped charge. So this flipping of charge is basically your nerve impulse moving down. So this is what your action potential is. Right, so this causes a reversal of charge, hence the inside of the axon now becomes positive in comparison to the outside. Right, this is how the action potential and the membrane has become depolarised, and it's swapped charge, so it's almost become repolarised in the opposite direction. So remember, whenever we're talking about charge, right, even though we're not talking about the movement of electrons, we can still talk about this in terms of a localised electrical current being set up. So the localised electrical circuits established by the influx of the sodium ions causes the opening of voltage-gated sodium ions a little further down. Right? So because this is voltage-gated, that change in the voltage there causes this sodium channel to open up. Right? This a little further down the membrane. So, if this sodium channel is opened up, then sodium ions can go and move through. So now, that part of the membrane, right, remember, when we're talking about it being like depolarised and flipping charge, is not happening on the entire nerve cell, right? Remember, this is like a tiny, tiny, tiny section because we're talking about individual proteins. So now, this side, or this part of the membrane, is flipped charge, right? So that depolarisation, so that flipping the charge, is being propagated down the nerve cell, right? So it's moving down, and it's being caused by the change in charge in this point here, causing your sodium channel to open up at that point there. Right, we don't want this depolarisation to stay there forever, right? Because that'd be ridiculous, because then all your nerve cells will all be on all the time, and you'd just be like, then you'd die. So, behind this region, behind this new region of depolarisation, so this is where we've gone and we've got this depolarisation occurring, voltage gated potassium channels open up. Right, because the potassium inside the axon is a lot greater than outside, the potassium ions diffuse out. Right? So, as the potassium ions diffuse out, we now end up back at this point here with more positives on the outside than on the inside. So now that takes us back to our resting potential, where the inside is negative into comparison to the outside, because we've got more positively charged ions on the outside than the inside. Right, these sodium gates, right, will close, so no more sodium can pass through, but we want to go and get back to that resting potential. So same as we talked about when we were talking about the maintenance of the resting potential, these sodium ions that we've got left in it, because now we've reached that area of hyperpolarisation, right, so it's taking it down to like minus 75 millivolts instead of minus 70, right? So we've got a lot more positively charged ions outside of the membrane in comparison to the inside. Remember, at this point here, when we reach hyperpolarisation, that sodium-potassium pump will pump out the sodium ions back to outside the membrane, and at the same time, it will pump the potassium ions back to the inside of the membrane, so we get back to that resting potential that's at minus 70 millivolts.